Welcome to Life Sciences Decoded, where we dive into the technology shaping the future of life sciences and healthcare. I'm your host, Susan Najjar, and today we're exploring the rapidly evolving world of RNA therapeutics. We are at a turning point in medicine where RNA therapies hold the promise to treat everything from cancer to chronic inflammation. But one challenge remains. How do we deliver these powerful molecules safely and effectively to the right tissues? Today, no orally delivered RNA therapeutic has yet been developed. In this episode of Life Sciences Decoded, we speak with Nigel Theobald, CEO of N4 Pharma, about a bold new approach, a delivery system that targets disease at the cellular level and is capable of oral delivery that could reshape how we think about RNA therapeutics. Nigel says RNA therapeutics are only as effective as the systems that deliver them. Welcome, Nigel Theobald. Welcome to the podcast. I'm very excited to be speaking with Nigel Theobald today. In many ways, COVID brought to the forefront how RNA therapies can be used to treat disease. And many biotech and pharma companies are racing to develop life-saving treatments for patients utilizing gene therapy. Nigel, it's wonderful to have you on the podcast. Welcome.
Thank you very much, Sue. It's great to be here. I'm looking forward to it.
Yeah, it should be a wonderful conversation. So before we talk about your quote around the need for RNA therapeutic delivery systems, would you please discuss the fundamentals of RNA therapeutics, what they are, how do they work, some of their benefits?
Absolutely, yeah. So I'll give you a bit of overview. I'm not a scientist, sorry. So when we talk about these things, yeah, I'll talk fairly generally. So RNA, and that's along with DNA, they're called what's new called nucleic acids, and they're one of the four major components of all human life. And then in effect, they're like the intermediaries. So what they do is they translate the code from a nucleus in the DNA, to tell the cell to produce a particular protein which the body needs to operate. And in things like cancer or irritable bowel disease and viruses like you mentioned, COVID, what you see there is you get the body producing more or too much or too little of a particular protein. So what the RNA therapeutics are there to do is to mimic the naturally occurring RNA in the cells, and that brings about a chance for a new class of drug that can alter the DNA coding. So these new drugs tell the coding in the cell to produce more or less of a particular protein. But for them to work, though, the RNA therapeutics have got to be delivered into the right cells. And that's one of the major sort of challenges that these systems still face today. But when you get it right, basically what RNA therapeutics mean is you can target any drug. Because you can just target anything because every cell is made up of a piece of DNA or RNA. So you can address previously undruggable targets such as transcription factors. And RNA therapeutics are also more cost effective to discover and develop than antibodies or small molecules. So RNA, it can be easily modified and quickly modified to create personalized treatments as well for individuals, or also keep up with evolving viruses like it did with COVID.
Yeah, that's fascinating. You commented that RNA therapies hold immense promise, right, for the future of personalized medicine. Would you please explain the roles of mRNA and siRNA?
Yeah, sure. So mRNA, which stands for messenger RNA, and siRNA, which stands for small interfering RNA, they're the two most prominent types of RNA therapies. So what happens with mRNA in the cell is that the mRNA it goes into the cytoplasm, so it's delivered there via whatever means possible, and then it tells the cell to make a specific protein, and it sort of controls. It turns on the production of a protein to make something that the body actually needs. And what you can do there is RNA therapies involve sort of one or two areas. One is replacement therapy. where you're putting in a particular RNA to replace it. Maybe you're replacing a piece of RNA that's gone wrong. You might have a cell that's producing the wrong type of something, and you put in a new piece of RNA that basically replaces that bad one and gets the cell working again. Or in the case of like vaccines, where you put a little bit of mRNA in, which is a small part of the particular, like the virus for the SARS-CoV protein, which basically it prompts the body to produce more of that protein so it can fight the virus. On the other hand, with siRNA, they work the opposite way. So instead of producing protein like mRNA does, they block the protein. So they bind with something called a risk factor, and it combines with it and stops the mRNA from being produced in the cell. So you're turning off. And particularly, that's useful in things like oncology, where the cell is producing way too much of a particular protein and therefore growing. You can use siRNA to block and stop and what they call knock down the production of that protein.
Okay, perfect. Yeah, well, it's interesting how they both interplay, right? And you've talked about the benefits and really why the potential of RNA therapies are so vast, right? But what are some of the biggest hurdles in drug development?
Um, the big hurdles for especially for RNA therapies is to get the RNA into the right cells safely and effectively, and that's quite a challenge. First off, because RNA itself is very vulnerable to degradation. So once you put it inside the body then says, "Oh, don't like it here," it's out to get it, and it starts to attack it straight away. So it does need some element of protection and stability. Let's assume though you can protect it. You then got to get it into the cell. So the next challenge is getting it across the membrane of the cell wall. And the issue there is that the cell is negative and the RNA is negative. And imagine it's like two polar opposites. One way, they bounce off each other. So it's really difficult to get through. So what people end up doing is putting a huge amount in because only a tiny amount gets into the cell. So what you then need is you then need a delivery system to get it in. But once you've got it in, you then have another challenge, which is it's in the endosome of the cell and it needs to escape from the endosome and go into the cytoplasm. Because until it can get into the cytoplasm, then it can't produce the protein in the case of mRNA or knock down the protein in the case of siRNA. Another aspect of problem that these hurdles for these systems are is targeting. If you just put RNA in, it'll just go all over the body. So you need to be able to target specific cells. And what we have today, primarily, we have things that target the liver, and that's because the delivery systems that are out there just go to the liver. But what you want is you want to be able to target a specific cell because you don't want to be turning off the RNA production in good cells around the body. You want to be just focusing on the ones where it's going wrong. So one of the potential side effects of these treatments is you can stop your body doing things that it should be doing while you're trying to address the things that it shouldn't be doing.
Right.
Another thing is that they're all injectable at the moment, and that's because it's very difficult to actually get it into an oral application to protect it sufficiently. So you can put it into an oral application, get it through the gut wall and into the stomach and into the gut. And two reasons there. One, the lipid systems that people currently use, they're lipid by nature, and those fatty acid problems that get broken down when they get into the gut. So they won't protect the payload at all. And the second delivery system that most people use are viral vectors. And the problem with these is they're very toxic and they cause a lot of unwanted side effect issues once they get delivered into the gut. Solve those two issues and you can start looking at oral delivery, which means there's far more application for these RNA therapies than just injectables.
Fair enough. Yeah. Well, and I think,
Sorry, I was just going to say,
yeah.
so there's actually a significant unmet need for a better RNA delivery platform, one that is targeted to specific cells, that both non-toxic and effective, and it's easily manufactured. And if it can be orally delivered, well, that brings in additional benefits.
So this last point clearly addresses the heart of your quote, right? RNA therapeutics are only as effective as the systems that deliver them, and why N4 Pharma has developed Nuvec, which utilizes a silica-based nanoparticle approach. What distinguishes it both biologically and commercially?
Okay, so I'll just describe it a little bit. So our silica nanoparticle is a tiny little nanoparticle with pores with holes, and it has this spiky structure on the outside. And that spiky structure is used to trap the RNA with its structure. And I don't know if you ever go out walking. In the UK, we do a lot of that. And if you go walking through the grass, you get these tiny little grass spurs that get stuck to your clothing. That's what our Nuvec system looks like. It's got this spiky structure that's very sticky. And that gives it lots of different advantages because it can deliver multiple things within the spikes. It's really easy to make. It protects the payload, and it allows us to go on to look at oral delivery. And I'll talk about each of these a little bit more. So one of the key things I mentioned is protecting. So it can shield the RNA from degradation because the RNA gets stuck and trapped within that spiky structure, doesn't get released, and it doesn't get attacked by the enzymes that are in the body once it's put into the body. So that's enabled us to look at both injection, but also oral delivery, something I mentioned earlier that I think is really crucial for RNA therapies. The other thing our system does is it's very good at that second one. We get it into the cell very quickly, but more importantly, we then release it from the endosome. Remember I said if it gets into the cell with a negative thing, it then has to get out of the endosome and into the cytoplasm. And existing systems like lipids have got really poor endosomal release. As low as 1 to 2% of the drug actually gets released into the cell to work. So again, a huge amount has to be used. We've got much, much greater endosomal release, which means we can use less drug and we can have more effective treatments being developed because we can get more drug into the cell. The other area that we look at with Nuvec is it's actually, because it's silica, it's very easy to make. It's not complicated like viral vectors and lipids. It's low-cost manufacturing. It's very scalable. And silica is sand. It's very, very widely available and very easy to manufacture. And the final thing that we have that distinguishes us from these other systems is because of that spiky structure, we're able to load, and we're easy to load it as well. We can put 3, 4, multiple different RNA onto one nanoparticle that each of them get trapped within that spiky structure. Whereas a lot of these other systems, they form by encapsulating something. So you can take one thing and you can put something around it, but you can't do that with everything. Whereas ours, you're just mixing it and you're putting multiple things on at the same time.
It's really helpful to have you explain it in that fashion, and I can see the benefits of how this delivery system really is impactful. Are you able to share an example of how N4 Pharma is utilizing the Nuvec delivery system to improve clinical use of RNA therapies?
Absolutely, yeah. So what our first product that we're bringing through clinic at the moment is trying to showcase all of those multiple different benefits, the protection, the oral delivery, multiple loading, targeting in nature. And our product we call N4101, great title, it's the first one we're doing. And we're looking at it as a therapy for inflammatory bowel disease, otherwise known as IBD, mainly ulcerative colitis, one of the key areas of IBD. But it's an orally delivered treatment, and it's designed to target the macrophage cells within the gut, because it's the macrophage cells that go wrong that cause the inflammation that in the disease of IBD. And the other thing we do is we're combining 2 different therapies on a single Nuvec nanoparticle. And as I mentioned, that's very unique for our particular system. So the first, we have an siRNA. And as I mentioned earlier, the siRNA is used to knock down something. And in this case, what it does is it knocks down something called TNF-alpha. So TNF-alpha is a protein, and in IBD patients, there's way too much TNF-alpha that's produced, which causes inflammation in the gut. Really nasty condition, IBD. So it's an overproduction of that inflammatory agent, TNF-alpha. And the body then has some cytokines in there, sort of naturally performing anti-inflammatory agents, one of which is IL-10. So what we do is we put an mRNA into the particle as well that boosts IL-10. So we've got 2 things. We've got an mRNA producing more IL-10, and we've got an siRNA knocking down the TNF-alpha. So we're stopping the production of TNF-alpha, and that TNF-alpha that's already been produced, we're causing it to be basically negated by the IL-10. So it's this dual approach. And then we're doing all that again by adding a ligand to target a mannose ligand, which is targeting the macrophage cells. And that ensures better targeting and better uptake of the macrophage cells, which are the ones that are causing all the issue.
Yeah, that's fascinating. I think so many people listening to the podcast can really relate, especially with irritable bowel syndrome being such a common ailment and some of the more serious forms of it really being in fact impactful. So are you able to share what data you have that demonstrates these benefits that you've, just described?
Yeah, absolutely. So just to point out, we are still in the preclinical phase at the rather than in the clinic, and we've got both in vitro and in vivo data. So in vivo is when we're doing animal work, and in vitro is when we're in the lab. So in the in vitro work, we've been very successful in demonstrating that our N4 formulation will actually both simultaneously reduce the TNF alpha and increase IL-10 production. And that we've also seen that when you add that mannose ligand to it, it greatly increases the uptake in the macrophage cells that we're testing it in. So what we've then done was we've then taken a formulation based on that data and we've put it into some in vivo animal work using a standard industry IBD model. But in this instance, we put it in an oral capsule. So we've taken the Nuvec formulation, we've put it in an enterically coated capsule, and enterically coated capsule means it's got a level of pH control on it. So you swallow it, or in this case, the animal swallows it, it goes into the gut, and it only releases the payload when that pH is at the right level. So it releases the payload actually in the upper GI tract in this case. But because we've got both the RNA, mRNA and the siRNA and the macrophage targeting, we've seen some really impressive results. So all key disease indicators for IBD and colon length, because IBD causes the colon to shrink. So if you have a good colon length, then it means you don't have IBD. So all the disease indicators showed that there was very, very reduced amounts of inflammation, and importantly, a total knockdown of TNF alpha, the bad protein causing all the inflammation. And we saw a greatly increased level of IL-10, both in the intestines and in the blood. And even we saw that after 15 days, showing that there's a clear sustained effect. So we're really pleased with the early results that we've had. But it's just to mention though, yeah, N4 is oral, but our delivery system isn't just oral. You can inject it subcutaneous, intravenous. But one of the extra benefits of the protective nature is that we can actually deliver it orally. So that's one of the areas why we're focused. So we're able to actually deliver something into the gut for localized treatment rather than injecting a lot of it, hoping that it will then make its way to the gut at some point.
That's amazing. I'm so looking forward to, your future results because it sounds like this delivery system is really able to do multifaceted, aspects of it and truly beneficial to patients who are suffering from which can be quite debilitating effects of that. So looking forward to hearing more about what you come up with and next steps and more data. So how close are we realistically to a future where RNA-based treatments are as common as antibiotics? And how central is delivery innovation to that transformation?
Well, that's a really good question. I mean, I've got no doubt, right, that RNA therapeutics have got an enormous role to play in the coming years. But to do that, you're going to have to solve the delivery challenges, as I mentioned before, which is one of the key focuses of our technology and other people out there. But if you can solve that, then, as I said earlier, you can target you can have a drug for any target because every cell has got RNA and DNA in it. So whatever disease, however common, however rare, you can come up with a therapy and a treatment. And if you can make it an orally based treatment, then you can start seeing how you can take it as common as you would antibiotics. But to get there, you've got to solve the delivery issue. And, let's be realistic, we're a long way from that at the moment, because at the moment, the treatments just go to the liver and they're injectable. And in order to be widely used as antibiotics, you've got to get it into a format that everybody can take in everyday usage.
Yeah. Well, and that's why you're developing Nuvec, and really excited to hear next steps on it. So we like to end on a myth buster. Many
Okay.
people believe, and you've kind of addressed this one already, that all RNA therapies need to be injected. Is this true?
Well, you might guess that I will actually turn around and say no. I believe that that is false. So a lot, all the drugs, pretty much all the drugs today with RNA are delivered by injection. They don't have to be. They're delivered by injection because of the stability issues and the delivery challenges that they face. But oral or topical delivery as well, where rubbing it on, onto the skin, is becoming more and more possible with new technologies like the nanoparticles that we and others are producing. And that's what we're working on. So whilst at the moment, there's no cure out there for medicines, this is where things are moving. So you take the small molecule traditional approaches where you're just treating a symptom of a disease because the IBD is a good example. You might be producing IBD, you've got inflammation, you're just treating inflammation. What you're able to do with RNA therapeutics, you're able to cure, you're able to stop that production, you're able to stop things, you're able to actually treat the disease and cure things. So we're looking in a future where RNA therapies can be used to treat to cause these, to treat diseases and cure them rather than just look at them messaging and massaging the systems from something that's actually gone wrong in the body.
Fair enough. Yeah, absolutely. It is so clear that we are at a turning point in medicine with RNA therapies. I can't thank you enough for sharing your thoughts and how N4 Pharma and Nuvec are a key driving force. It has been a real pleasure speaking with you, Nigel. Thank you.
Likewise, thank you, Sue. It's been a pleasure, and hopefully, I've got a few points across to try and make things a little bit clearer in a not always too scientific way. Thank you.
Absolutely. No, thank you so much.